Okay, what's an operon? So an operon is a segment of DNA containing a group of genes that are transcribed together. So it doesn't exist in eukaryotes. Really keep this in mind, operons only exist in prokaryotes, okay? So it's a bunch of genes which are transcribed together. They separate from the promoter where the RNA polymerase binds. So an operon consists of a series of structural genes which are transcribed together, only in prokaryotes. And so one method of gene expression regulation is a trip operon, and this is explicitly defined in your study design. There also used to be something called the lac operon, so I'd recommend looking that up as well. Lac operon and trip operon, but now your study design is going to be testing the trip operon, and so we'll look at that soon. So there is a gene in some bacteria, including E. coli, and this encodes for the production of the particular protein tryptophan, also known as TRP. When too much tryptophan is present, we have tryptophan binding to a repressor. What happens is that this tryptophan molecule binds to the repressor and activates it, and once it's in this activated conformation, it can then bind to the operator region of a gene. And this will prevent transcription from occurring and prevents RNA polymerase from binding to the promoter region. So when you've got too much tryptophan, it's kind of like a defense mechanism kind of thing. It's going to kick in and be like, look, too much tryptophan, let's activate the repressor. So it activates the repressor binds, and it prevents transcription. When there is little or no tryptophan, this means that tryptophan doesn't bind to the repressor, and it causes the repressor to actually change in conformation. The repressor will then detach from the operator region, and this allows transcription to occur, as RNA polymerase can then bind to the promoter of the operon. The repressor is coded for by a regulatory gene, TRPR, and this sits upstream, it's just like way out, separate from the operon. And so it's way upstream of the tryptophan producing gene on the operon. So the repressor is also a protein or an enzyme. It's encoded by a gene because it is a protein, and it's just like separate to the entire operon. Okay, diagram. So this line here, it took me a really long time to know this or learn this, but that means it's like way away. Like it's not right next to it, it's actually like quite a long way away. So here is your operon. Trip E, D, C, B, and A are our structural genes. And then you've got a few regulatory genes. And you've got the promoter region where the polymerase wants to bind, and the operator region where the repressor will bind. And here you've got TRPL, which is known as the leader sequence. And we'll look at that soon. TRPR is way upstream. It encodes for the TRP repressor. And so what happens is that will be transcribed into mRNA. That mRNA will be translated into an actual protein. And so what that protein is actually the TRP repressor. And so here it is in its inactivated state. When you've got a high amount of tryptophan, it will bind to the repressor and activate the repressor. After this, like when the repressor is activated, it's actually going to bind to the operator region. And so in binding to the operator region, it causes a physical blockade there. And this actually prevents the RNA polymerase from being able to bind to the promoter region. As a result of this, the RNA polymerase can't actually go through and transcribe these structural genes. So you have kind of the entire operon just like being shut off. Like these genes are going to be downregulated because the polymerase can't actually bind the promoter region and can't actually go through and transcribe these genes. Okay, is it still recording? Oh my gosh, I thought it stopped recording for a minute. We are fine. Okay, so that was the first method of kind of like regulating the trip operon. So you actually have the repressor being activated. When the repressor is activated, it will bind to the operator region and prevent the polymerase from going through and transcribing these genes. We have a few more methods of regulating trip operon. So one of them is known as attenuation. And so, like, although repressors prevent transcription from starting, attenuation actually prevents transcription from completing. And so a region called the leader codes for an attenuated sequence. And the attenuated sequence forms hairpin structures. So within the leader are two tryptophan codons. This means that in order to translate the leader, we need tryptophan. This is kind of a complex process, so I'll try and work through it slowly. I mentioned we've got the TRPL here, which is the leader sequence, and inside the leader we've actually got an attenuator. So this attenuator forms a hairpin loop. So I might just open up a browser here and explain what I mean by that. Shouty capitals. Hairpin loop. So RNA we know is single-stranded, unlike DNA, which is double-stranded. What happens is that RNA can actually form loops in on itself. So you can see here that while it's a single-stranded structure, it's forming complementary base pairing with its own strand, right? And so it's forming lots of loops. And so as a result of this, you have the formation of hairpin loops. So what happens is that the TRP sequence, which is the leader sequence, it will actually be transcribed into mRNA. Consequently, this mRNA will then actually go through and it will form some hairpin loops. And there are two types of hairpin loops. Something else to note is that within the leader, there are two tryptophan codons, and these codons encode for tryptophan protein. So in order to translate the leader, we need tryptophan. And so depending on the level of tryptophan we will have different hairpin loops forming. How are we going for time? We're fine. So in the case of low tryptophan levels, the ribosome, okay, I need to pause here. I didn't learn this until uni. This wasn't part of my study design. I had something called the lacquer one, which was much more simplistic. I found I had a lot of trouble learning this because it just took me, I don't think anyone really simplified this enough for me, but I didn't realize that in prokaryotes, transcription and translation can occur at the same time. So I show my students a diagram. So you can see here, you've got the DNA. In prokaryotes, there's no nucleus. As a result, transcription and translation can occur together because there's no segregation of these two processes. Because the nucleus means the ribosomes can't get in, and the mRNA's got to leave the nucleus first in order to be translated in eukaryotes, right? But in prokaryotes, there's no such thing as a nucleus. Sorry. Just a drink. So because in prokaryotes, the DNA and because the DNA isn't sequestered in the nucleus, you can actually have ribosomes go through and translating that mRNA straight away. So what you do is you have RNA polymerase reading the DNA and making mRNA. So this purple thing here is the mRNA strand. And pretty much as soon as it starts reading it, a ribosome is going to jump on and start translating that, okay? So pretty much transcription and translation are occurring simultaneously because there is no sequestering of the mRNA in the nucleus because there is no nucleus. So it's really important to understand that transcription and translation occur at the same time in prokaryotes. Okay, so what happens is that you have that leader sequence being transcribed and it will form some kind of a hairpin loop. And if you've got enough tryptophan, you can translate the leader. What happens there is in low tryptophan levels, the ribosome will move through the leader really slowly as we need tryptophan for translation. As such, a hairpin loop is created that does not stop transcription, so transcription occurs. In high tryptophan levels, the ribosome will move through the leader very quickly as we have lots of tryptophan to translate the sequence. A hairpin structure is created that does not stop transcription, therefore transcription doesn't occur. So it does stop transcription, so therefore transcription doesn't occur. And as such, the ribosome falls off the mRNA and RNA polymerase detaches from the operon. This is all really complex. The way that I think of it is if you've got low amounts of tryptophan, when the ribosomes go through it, they've got to translate it slowly because they're looking for tryptophan. They've got to go through and search for that extra tryptophan, right? So it's kind of a slower process. So what happens is they go through and they read the mRNA slowly. And they read it very slowly. So they're quite calm and relaxed. The way I think of the other process though is in high tryptophan levels, you can quickly translate, you zoom through it, and it kind of creates a formation of a hairpin loop, which in my head I like to think of it as it zooms through super fast, it causes the ribosome to fall off. It just goes through, zooms so fast, it falls off. And it kind of disturbs the RNA polymerase, and so this causes the RNA polymerase to also detach. So this will stop transcription, and transcription will stop occurring. I don't know if that makes a lot of sense, so I'll just try and say it again. In low tryptophan levels, I like to imagine that the hairpin loop, it's quite a slow, cruisy process. The ribosome go through and translate it slowly. This doesn't disturb the RNA polymerase at all because it's going through and translating it very slowly, very carefully. In cases of high tryptophan levels, the ribosome will zoom through that loop and translate it very fast, and as a result, you're actually going to have the ribosome falling off. And I just kind of think of it's called like an earthquake, and it causes the RNA polymerase to also fall off. Therefore, transcription will be halted as well. It is quite a complex process, and what I really recommend doing is looking up the VCEE Bio FAQ document. So if you look up this document, it really discusses what you need to know about this tricky concept that is, oh, it's PA, sorry. Why did it not download? Okay, it did download. This tryptophan, or trypoperon. I think the trypoperon is probably the most complex process we'll discuss. That and CRISPR, I think. This might be a bit harder, I think. So definitely go through and look at this VCEE FAQ document because it really explains what you need to know about the trypoperon. You can see that diagram is literally taken from this document here. It explains what you need to know about it. And it kind of goes through lots of really difficult areas of study or like topics that you need to know. And so making sure you read this and understanding what goes on is really important. So do recognize that tryptophan or the trypoperon in prokaryotes has transcription and translation occurring together at the same time, and it's really important that these processes kind of work together. And so if the ribosome falls off, this can actually alter the polymerase. The polymerase may also fall off too. Whereas if the ribosome goes through and translate it very slowly, then that polymerase can go through and transcribe it slowly and it won't fall off. Cool. So just kind of emphasizing here that attenuation is possible because transcription and translation occur at the same place in the cytosol. And this isn't possible in eukaryotes because they don't occur in the same place because you actually got the presence of a nucleus, meaning that transcription must occur in the nucleus and translation occurs in the cytosol or the RAFI. So as RNA polymerase moves to the operon, the ribosome can begin translating the mRNA even though the full strand isn't completed yet.